All right, we are going to talk about our next lab, which is entitled, If You're Not Part of the Solution, You're Part of the Precipitate, and we are studying solubility trends on the periodic table. So just like we can study the trend in atomic radius, the trend in ionization energy, and the trend in electronegativity, there are other properties that have trends as well, and one of those is solubility, and that's what we're going to be looking at in this lab. So the way that we identify chemical reactions is through visual observations in this class. So how do we know if a chemical change or a chemical reaction has occurred? We're going to look for a couple things. One indicator is effervescence, which is just a fancy word for bubbles, and that's seen here with that green backdrop. We also will be looking for precipitate. Precipitate is when a solid forms inside of two solutions, and that looks like this with the blue background. You can see all the little solid particles forming in that liquid solution. Odor is an indicator of chemical change. This fellow in the tie-dye lab coat is demonstrating wafting, in which case you gently fan any fumes towards your nose. We're not going to be doing a lot of wafting in this class. There's probably not going to be a lot of chemical changes that we can detect via odor because many times if you create something that has an odor, it's a noxious gas or a poisonous gas, so we try to stay away from that. We look for color changes. So for example, if you mix these two solutions, we've got a transparent yellow solution and a clear colorless, transparent colorless solution. If you mix them, it turns the solutions red, which is really interesting. So we look for color changes like that. And then the last indicator is heat. And because we do small scale chemistry, we're really only dealing with one little drop of each chemical solution from a pipette. We usually can't detect a heat change. However, changes in heat are one of the most obvious types of indicators on larger scale reactions. So if you think about those little heat warmers that you use, like if you're skiing, those little packets and you shake them and they get warm, that's a chemical reaction. And so that heat is indicating that a chemical change is happening inside of that packet. The way that we can easily remember what these are, what these indicators are, is with the acronym EPOC. And it sounds like a made-up word, and it is, but it kind of helps us be able to remember those 5 things that we can look for to detect a chemical change. Today we're really focused on solubility, so I want to talk about what solubility means. And solubility is the ability of a substance to dissolve in water. So we sort of intuitively, based on our life experience, might have a feeling for this. If you put sugar in water, it dissolves. If you put salt in water, it dissolves. If you put a piece of glass in water, it does not dissolve. If you put a rock or a piece of sand in water, it doesn't dissolve. So that's the real difference. And what we're going to be observing is when you take two solutions that are soluble, so it doesn't look like there's anything in them necessarily. They're typically transparent. They may or may not be colorless. But then when you combine them, a suspension or a precipitate forms. This indicates that there's a chemical change because a new insoluble substance has formed. So you've taken two things like salt that can dissolve in water, and when you mix them, the elements recombine to make something that cannot dissolve in water. And in this figure to the right, we have an example of a solution. You can't tell that there's anything in it. It just looks like a liquid. A suspension is going to show some little particles of insoluble stuff and maybe start to form a little pile at the bottom of a test tube. And a precipitate is going to be all the way settled at the bottom of a test tube. Now for us, because we're using small scale, again, we're going to have like 2 drops of solution that we're looking at. That is going to mean that we will be seeing some maybe suspensions or precipitates, but they'll typically look just opaque, right? And if you use a toothpick to kind of spread them out over the reaction surface, then they're going to start to look more speckled, and you might see more of that suspension. The families that we are studying are the alkaline earth metals, so that's second column, shown here in green. We're also going to study the halogens, which we sometimes call the halide ions, and that's fluorine and chlorine and all of those guys in the second to last column on the periodic table shown here in yellow. So we're going to be looking at those 2 families to look for a trend in solubility. Because they're on opposite ends of the table, if we can find a trend that works for these alkaline earth metals and a trend for these non-metals, if that trend is the same, then it might be okay to make an assumption that the solubility trend is uniform throughout the whole periodic table, if it is true for our metals over here and our non-metals over here. So that's kind of the assumption that we'll be making. And maybe that won't be a good assumption. We'll see what the data says. So thinking about our planning for this lab, the purpose is to discover a trend in solubility based on studying the alkaline earth metals and the halogens. So we might find that there is no trend in solubility. We might find that there's a different trend in alkaline earth metals than there is in halogens, or we might find that they have the same trend. And so that is what we're trying to discover today. And the essential questions that we'll be looking at are, what is the solubility trend for each group? You should define and explain all the periodic trends we've talked about so far, and speculate as to how or why they do or do not relate to solubility. Do you think that the atomic radius of an element might relate to its ability to be dissolved? Do you think that its electronegativity might relate to its ability to be dissolved? So you're going to think about those things and kind of hypothesize whether or not you think they are related. Using logic. And then you're going to develop a hypothesis based on the other periodic trends. So you're wondering which periodic trend might account for solubility, and based on those things, you're saying, well, I think, alkaline earth and halogens are going to have the same solubility trend, or I think they're going to have opposite solubility trends, or I don't think there's going to be a trend in solubility for either group. Those are the types of things you'll be hypothesizing. For the procedure, basically you'll have a pipette, and the pipette is going to have the label of the type of chemical, and you'll put 1 drop in the center of the X. So you'd find KF, you'd put 1 drop in the center. You'd find calcium chloride, CaCl2, you'd put 1 drop in the center. And then you could use like a toothpick or a cotton swab to kind of smear those together to make sure the full reaction happens, and you're going to be looking for what occurs. So you're looking for all those epoch indicators, and in particular, you're really looking for that precipitate suspension thing, because that indicates solubility. If there's a chemical reaction, but it doesn't relate to solubility, we still want to notate that it happened, but we don't really care as much because we're really specifically looking at solubility. So you should record the appearance of the solutions prior to mixing to establish a point of comparison. If you're like, oh, I mixed CaCl2 and KF and it was blue, that's not very meaningful if it was already blue, if CaCl started out blue, right? So you want to have that point of comparison. Do not ever touch the tips of the pipette to the reaction surface or to the previous drop or to the tip of another pipette. This type of contamination will ruin the entire contents of the pipette, and so you want to be really careful about that. Then, like I said, you can use a toothpick to mix the drops and observe any precipitate or suspension. For your data collection, you're going to make 3 data tables, probably. You want to have a list of what the reactants looked like before the reaction, so what they look like while they're in the pipette. You want to have a table for alkaline earth metal reactions, and you want a table for halogen reactions. And this is, over on the right, is what your reaction surface is going to look like with the X's. So you can just make your data tables like this, without the giant X in the middle, and then you would write your observations in this box. You can write the before observations in this box. Instead of making 3 data tables, you could write the before and the after. It's whatever you want to do, but you need to have that before and then the description of what happens after they're mixed. These observations are going to be qualitative, and I expect you to be very descriptive. So that would look like this. After mixing the solutions, it became bright yellow and opaque, right? It was see-through. It wasn't transparent anymore. With flecks of light yellow precipitate visible after spreading the sample using the toothpick. And so that's really, really specific. It's showing a color change because it became bright yellow, showing that it became opaque, and that indicates some type of suspension or precipitate, and then after you were able to spread it around, you could clearly see a yellow precipitate. And this is just a little note about what I said earlier. There's many indicators of chemical change that you should document, but the most important one for this lab is going to be the presence of a precipitate, because that indicates that it's not soluble. Okay, for data analysis, consider making 2 tables again to summarize the solubility of the ions. So, solubility of each of these ions, magnesium, calcium, and strontium, and then fluorine, chlorine, bromine, and iodine. And then you will state the overall solubility trend for a group. So you could really just make tallies and just say, okay, magnesium had, precipitated 3 times, or calcium made a precipitate twice. And then you can try to state an overall trend for each group based on that information. For your evaluation, you're going to revisit the purpose of the lab. So what were we looking at? Why were we looking at it? Briefly recap the procedure. Report the results. So you need to answer these questions listed here. What was the solubility trend for the alkaline? You have to use evidence from your data collection and analysis sections to support your claim. Then do the same for the halogens. And then which periodic trend, atomic radius, first ionization energy, or electronegativity might account for the solubility trend. And I want you to kind of justify or explain your answer using logic, right? So that it makes sense. You should report any errors or anomalies that might have interfered with your results. Why do you think they happened? How would it have changed your results? How could it be avoided? Please don't say things like human error. That's not really that meaningful. Okay, and we can talk more about that after the lab is done. Timeline for this B group, please read and annotate your lab sheet and rubric, and complete planning and set up data tables for your data collections before Tuesday, February 16th when we do the lab. A group, read and annotate your lab sheet and rubric.